Welcome back again. So let us move on to our next cell organelle, that is lysosome. So this is a very simple cell organelle. Actually there is no that much structural complexity. A very simple cell organelle in structure, but its function is very important. Okay. So what is lysosome? So lysosomes are discovered accidentally by a Belgian scientist D. Doo, full name Christian D. Doo, in 1955. Actually through a fractionization technique that he did in his laboratory. So these organelles are observed under electron microscope by Novikov. And Novikov named this organelle as a lysosome. This is a small body, actually no particular structure for this. And they are actually known as suicidal bag of the cell. Clear? So while this in the term itself you know what is suicidal bag. Okay. Then the lysis, the term lysosome. Lysis means what? Digestive or loose or digest or break, that is lysis. So breaking bag, that is lysosome actually. They are the small vesicles and that contain powerful hydrolytic enzymes in the form of crystalline or semi-crystalline granules. Okay. So they are actually a form of vesicle, a bag containing powerful hydrolytic enzyme. That's only the content. Clear? Now, important enzymes are acid phosphatase, sulfatases, proteases, etc. present inside this, and they are called acid hydrolases because these digestive enzymes are function as acidic medium, that is in between pH 4 to 5. They function in acidic medium only. Okay. It maintains the pH inside. Acidic conditions are maintained inside the lysosome by pumping of H plus ions. Then lysosome having a simple covering, and inside the cover hydraulic enzyme present, a bag or a vesicle, and large number of such enzymes, digestive enzymes are carried inside the bag. Okay. And another very important point is they are usually budded off or cut off from some other organelles like our Golgi complex or endoplasmic reticulum, etc. And the diameter of this cell organelle is between 0.2 to 0.8 micrometer. Okay. And interior is usually solid, means having a dense region and a central mass. So simply you can see the structure like this. I will show you. See. Nothing to explain structure. This is the simple circular outline, and in the center portion you can see it is somewhat like this central region, and two regions you can distinguish, dense region and outer region. Okay, they are the actually budded off. I already told you, budded off from endoplasmic reticulum or Golgi complex. They are the precursors or of certain reactions, and actually in these vesicles store enzymes and modified in the form of lysosomes. Okay. And these take part in the irregular digestion of various materials. So from outside some of the materials enter inside, that is by two processes, namely phagocytosis and pinocytosis. When liquid particle enter inside, that is pinocytosis. When solid enter inside, that is phagocytosis. So whatever it may be, when endogenous or exogenous origin, when substances enter inside, it will digest it. Clear? So extracellular digestion is performed by these lysosome in certain conditions. So intracellular digestion as well as extracellular digestion is carried out inside lysosome. Okay. Then lysosome passing through different stages in its in a particular cell. Different stages. Okay. So that's why. So they acquire different shape and different functions. So having different stages in a cell. So that's why they are known as polymorphic. That they exhibit polymorphism. This is very important question. Why they are known as polymorphic? Because they acquire different functions or different stages in a cell. So depending upon the morphology and its function, there are 4 types of lysosome recognized. What are they? Primary, secondary, residual bodies, and that autophagic vacuoles. Primary lysosome, secondary lysosome, residual bodies, and autophagic vacuoles. What are primary lysosome? They are the first formed lysosome. They are actually the budded off, pinched off vesicles from Golgi apparatus. Okay, and they fused with the endosomes and become functional. Then primary lysosomes in structure, they are very small and contain hydrolytic enzyme. They are very small. They are the first formed one, budded off from Golgi. Secondary are actually heterophagosomes, fusion product actually. Okay, it is formed by fusion of food containing phagosome with the lysosome. Phagosomes are vesicles, food containing vesicles fused with the lysosome. That is secondary. Okay, then after that fusion, what happens is in the digestion occurs. Then digested food passes into cytoplasm. Clear? So, food storing phagosome fused with the lysosome forming secondary lysosome. Then after digestion, food passes into the cytoplasm. They are later digested or settle as undigested. Clear? See, what is residual body or third one? Residual or tertiary. You can see these are the lysosomes that contain undigested food. Okay. These residual bodies or lysosomes pass outwardly and fuse with the plasma membrane and throw out to the debris into the external side. That's by exocytosis or ephagy. Means through the plasma membrane, these residual bodies are moved out to the cell and destructed. Clear? This is by exocytosis, by absence of hydrolytic enzyme. They are actually what? Residual bodies are actually the lysosome carrying undigested food material. Clear? Then autophagic vacuoles. What are autophagosomes or autolysosomes? They are produced by fusion of number of intracellular organelles. Lysosome fused with some intracellular organelle, and these primary lysosomes or secondary lysosomes etc. fused with other cell organelles and they destroy the cell organelle and themselves they destroy. This phenomenon is known as autophagic, means they are ingested or they are lost forever. The cell organelles are destroyed, other cell organelles are destroyed, okay, by its digestive enzyme. They self-destroy. That's why they are known as suicidal bats. They are autolysosome or autophagic vacuoles. So, primary, secondary, residual and autophagic vacuoles. Clear? Okay. Then, let us move on to function of lysosome. So, you know now, lysosome contain hydrolytic enzyme. So, very important function is intracellular digestion and also extracellular digestion. So, individual cell obtain food through phagocytosis, that is solid particle enter inside the cell, that is digested. Extracellular means they release enzymes outside the environment and digestion take place. Then body defense involved and autophagy, self-destruction, unwanted cell organelles are destroyed by lysosome. Then for example, it is applicable in certain organisms, that is metamorphosis in amphibians etc. It is applicable, removal of their tail, their gills etc. in the case of tadpole larvae etc. So, these are some of the events taking place. See, this is the Golgi complex. From Golgi complex, that is budded off is the vesicle. This budded off vesicle fused and forms secondary lysosome. What is the food left? That left food is known as what? Residual body. They enter in the plasma membrane. This is the plasma membrane and residual food particle expelled out by exocytosis. Clear? Then some of the cell organelles fused and form inside the lysosome, that is autophagic vesicle. Then food vacuoles enter inside, moving outside, liquid particles moving outside etc. So, lysosomes budded off from Golgi complex or from endoplasmic reticulum, primary, secondary, residual body and autophagic vacuoles. So, what is a lysosome? Actually, they are the bag. They are the vesicle filled with the digestive enzymes. And what are the different types of lysosome? Why they are known as polymorphic? And what are the functions of lysosome? Clear? So, I hope you understood well. So, have a nice day. Thank you.